Açayım hocam. Dear respected participants, we are continuing now with the next section. This section is going to be moderated by Professor Dr. Attila Bobis. And the first presentation is going to be done by dear Professor Attila Bobis. Are you here? Till Professor Dr. Attila Bobis come, we can start with this with the presentations. First presentation is going to be held by Dr. Delka Salkova. Her presentation title is Detection of Varroa Destructor Mite and Nosema SPP in bee samples from Bulgaria. Dear Doctor, welcome to the Congress. I ask a request for opening your voice, and you can make share your presentation.
Yes, hello, just a moment, share screen. Yes, thank you very much for the opportunity to take a part in your Congress. And I wish good luck and success of our participation. Greetings from Sofia, from Bulgaria. Now I think, okay, disable why. Something wrong. Share screen, yes. Host disable, participant screen sharing, why?
We are now trying to open it. Is it open now?
Yes, but it don't allow me.
Just a second.
Yes, now.
Now
Yes.
Yes, you can do
I
it.
think, yes. Fine. Yes, I think.
Yes, we can see your presentation now.
Okay. Dear participant, today I will present our works with Professor Kalinka Gurgova from private laboratory Primavet. We investigated some bee samples for Varroa destructor mite and Nosema species in bee samples from Bulgaria. Despite the small size of Bulgaria, the country is one of the most suitable places for honey production due to its diverse plant life and temperature climate. Everybody knows that in recent years there have been significant losses in beekeeping around the world. The beekeeping is facing a number of challenges. On the one hand, the impact of the increasingly unfavorable for the bees environmental conditions like climate change, lack of feed, insufficient variety of food, pollution of water, soil, and vegetation with toxic elements used in industry and agriculture, pesticides, etc. And on the other hand, the spread of new resistant pathogens, parasites, viruses. One of the main reasons for the high losses of bee colonies are parasitic disease. Varrosis and nosemosis are the prevalent and damaging invasive disease of honeybees, which cause large losses in beekeeping. The aim of our work is to present the results of the studied bee samples in a 2-year period during 2020-2021 for 2 of the main pathogens on the bees, exactly Varroa destructor and Nosema species, leading to reduced production, weakened colonies, and often high losses in bees. Material and methods in a 2-year period during 2020 and 2021 in private laboratory of Primavet and in Department of Experimental Parasitology to Institute of Experimental Morphology, Pathology and Anthropology, Bulgarian Academy of Sciences, were examined 185 bee samples from apiaries located in different districts of the country. To the private laboratory of Primavet were sent 94 samples for diagnostic, mainly from colonies with some pathological problems, such as abnormal behavior, the population of beehives, weakness, and high losses of colonies. In Department of Experimental Parasitology were investigated 91 bee samples for monitoring of varroa structure and Nosema species. The methods were used of OIE Terrestrial Manual, Chapter 3.2.7 version adapted in May 2021 for varroa structure identification, and OIE Terrestrial Manual, Chapter 3.2.4 version adapted in May 2030 for the identification of Nosema species, respectively. Diagnostic methods used to detect varroa destruction and to prove spores of Nosema species included morphological identification, white microscopic examination, bee examination by alcohol wash tests. Investigation for varroa destructor mites. The dead bees from the bee samples were flooded with industrial alcohol, stirred continuously for around 5 to 10 minutes. The mites that floated to the surface were identified and observed. Then the contents were placed of the sieve on a bright plate where the mites can be easily identified and counted. The percentage of infestation level was calculated by following formula. Percent of varroa destructor mite is calculated by number of phoretic numbers divided by number of adult bees and multiplied by 100. Investigation for spores of Nosema species. The abdomens of 30 bees of each sample are obtained in 30 milliliters of distilled water. Of the suspension are made smears on a glass slide. Smears were air dried, ethanol fixed, and were stained with Giemsa stain for 45 minutes. Nosema species spores had a distinctive appearance with thick instate walls and an indistinct blue interior without visible nuclei. To quantify the average infection level, spores were counted and were calculated per bee. Results from private laboratories showed that 32.98% of bee samples were infested with varroa destructor. The degree of invasion in the bees was in the range from 0.5% to 60%. Spores of Nosema species were established in 26.60% of samples, with a degree of invasion in the range from 10 to 5 to 40 to 26 million per bee. Mixed infections of both parasites were observed in 28.72% of sample analyzed. Negative samples were 11.17%. And these results are presented on figure 1. Results from 91 investigated samples in experimental laboratory showed the highest percentage of mixed infection, 36.26%. Samples invested with varroa structure were 31.87%. Spores of Nosema species was proved in 24.80% B samples. Negative samples were only 7.69%. And these results are presented on figure 2. We can conclude it that our study showed prevailing higher percentage of infested with varroa structure mites B samples than those positive for Nosema species. We should also note the relatively high percentage of samples with mixed infection. A very small percentage of the tested samples were negative for both of pathogens. Thank you for your attention.
Dear Delkova, thank you very much for your nice presentation. Due to the limited time, we will organize this session so we will get first all presentations. Meanwhile, the audience can write the question in the chat box. Afterwards, the moderator at the end of the session direct the questions. Thank you very much again. We will continue.
Thank you very much.
We will continue now with the next presenter. The next presentation belongs to Emine Sönmez. Dear Dr. Emine Sönmez. We have demanded to open your voice.
Hello.
Hello. Welcome. We are listening to you. We are curious about your presentation. Dear Dr. Otilla Bob is here. The moderation of the session is going to be done by Professor Dr. Otilla Bobis, very experienced person, academician in the bee products, and I'm giving the word to her. Dear Professor, we are now at the second presenter, Emine Sönmez.
Yes, thank you, Dr. Ulaş. I'm here. Sorry for my delay, and without taking the time from Dr. Emine, I give her the word.
Thank you. Hello to all my dear colleagues and participants. First of all, I would like to thank Associated Professor Ulaş Acarus and his team for organizing this valuable congress. Please let me to tell some information about chemical profiling, antimicrobial and antifungal activity of Anatolian honeybee venom. Pardon. Can you see my screen?
Yes.
Yes. There are many studies involving infections such as cellulitis, pharyngitis, folliculitis, and abscesses in the world, caused by gram-positive bacteria such as Staphylococcus aureus, Group A beta-hemolytic staphylococci, alpha-hemolytic staphylococci, and Group C beta-hemolytic staphylococci. On the other hand, Staphylococcus aureus is the main causative pathogen of impetigo, paronychia, and staphylococcus scalded skin syndrome. In complicated cases such as abscesses, necrotizing fasciitis, and diabetic foot infection, gram-negative bacteria such as Pseudomonas aeruginosa, Vibrio sp, Aeromonas hydrophila, and Escherichia coli are seen to be causative agents. Normal skin commensals such as Staphylococcus pyogenes and Staphylococcus aureus multiply rapidly during adolescence and are often involved in the development of acne. In addition, Staphylococcus pyogenes usually cause superficial infections of the sebaceous glands. Antibiotics such as ampicillin, cefazolin, vancomycin, tetracycline, and erythromycin are typically used to inhibit this kind of inflammation and kill bacteria. However, in a statement published by the World Health Organization, it is thought that these infections will pose a great threat to human life due to the lack of new antibiotics against drug-resistant infections. In addition, annual deaths attributable to antimicrobial resistance are expected to exceed cancer deaths by 2050. Due to the severity of the situation, there is a need to develop less toxic and more effective natural agents with different mechanism of actions against these microbial infections. In the way of finding new strategy, antimicrobial peptides, an important part of host defense family, emerge with tremendous potential. These peptides have some advantages like broad antimicrobial spectrum, rapid action, lower risk of resistance, low toxicity, and high selectivity. With these advantages, antimicrobial peptides have been widely investigated in both mechanism of action and new therapeutic application like anti-cancer, antimicrobial effect, wound care, sepsis treatments, and cosmetic ingredients. These peptides are isolated from natural sources such as mammals, plants, and insects. Apart from snake and scorpion venom, honeybee venom is known to have a natural antimicrobial effect and has long been used to treat a number of medical conditions. Since skin pathogens not only include just bacteria species, but also viruses, fungi, and protozoa, antimicrobial peptides with such a broad spectrum can be more useful compared to traditional antibiotics. Furthermore, due to its low ability to permeable into bloodstream, antimicrobial peptides preparations have the benefits of high concentration at target site for topical administration. Especially in wounds, antimicrobial peptides not only prevent pathogen proliferation and biofilm development in lesional skin, but also promote wound healing through modulating the cell migration, chemotaxis, cytokine release, and angiogenesis. Generally, the biological activity of bee venom originates from peptides, enzymes, amino acids, sugar, phospholipids, and volatile compounds. Bee venom is a mixture of substances that exhibit a wide variety of biological activities, including cytotoxic, hemolytic, and anti-inflammatory activity. Mellitus and phospholipase A2 are the most abundant component of bee venom, and these are considered to be responsible for many of venom's beneficial effects, including antimicrobial activity against a variety of microorganisms. Today, it is widely used to cosmetic applications like acne vulgaris, facial wrinkle, atopic dermatitis, psoriasis, and vitiligo. Considering the increasing prevalence of antimicrobial resistance mechanisms, breakpoints in antimicrobial susceptibility, testing guidelines are prone to change due to the detection of new resistance mechanisms. Therefore, the aim of this study was to examine the antimicrobial and antifungal activity of Anatolian honeybee venom against bacteria and yeast-like fungi that cause dermatological infection as an alternative potential. In this way, honeybee venom sample used in this study was collected from Hughes Induced University Beekeeping Research Development and Application Center using an electric shock device. At first step, to collect the venom, necessary equipment and devices were prepared, and then prepared devices were put in the Hughes. Since the electric current would encourage worker bees to sink into the glass plate, the bees released their venom onto the glass when they receive electric shock. The honeybee venom left on the glass was immediately brought to the laboratory without waiting and was collected from the plate with the help of a scraper. And then collected venom was put into the amber bottles, sealed in the airtight manner, and stored at -20 degrees Celsius. HPLC UV devices were used for detecting and calculating the chemical content of bee venom. For HPLC analysis of venom, sample was weighed and dissolved with pure water, then diluted and passed through a filter and given to the device. The work was done with a C18 column. The UV detector was adjusted to suitable valence and the peaks were scanned. On this slide, you can see the list of used microorganisms. A total of 13 different microorganisms were used, and 3 of them were gram-positive, 7 of them were gram-negative, and 3 of them were yeast-like fungi. All strains were supplied by microbiology laboratory collection from Recep Tayyip Erdoğan University. For detect the antimicrobial potential of microorganism, we used 2 methods. At first step, agar-well diffusion. Second one was minimal inhibitory concentration dose. For agar-well diffusion method, at first, McFarland's adjusted to 0.5 and then microorganisms were separated on the appropriate media. Later, wells were punched in each plate using a sterile borer. Bee venom was prepared in 4 different concentrations and then filled into the wells in amount of 50 microliters. After that, petri dishes were incubated for 24 and 48 hours for bacteria and yeast, respectively. And then the inhibition zones were measured with the help of a caliper. After agar-well diffusion method, minimal inhibition concentration of Anatolian honeybee venom was determined by broad deletion method. We did 2-fold serial deletion of honeybee venom in macro deletion plates. And then we prepare McFarland at 0.5 again. Later, we dispense bacteria inoculum in macro deletion plates with diluted venom. After incubation period, we detect the mild values. For results, in this experiment, chemical profile and antimicrobial activity of Anatolian honeybee venom were determined against dermatological pathogenic microorganisms, including gram-positive, gram-negative bacteria, and yeast-like fungi. First step, HPLC method was used for chemical analysis of the venom. The HPLC studies demonstrated that a very good separation of apamine, phospholipase A2, and mellitus were obtained. The result of the HPLC analysis confirmed that the examined honeybee venom sample was of a good quality with a mellitus content of 57.62%. Because we know that in generally, mellitus content of bee venom is ranged from 40% to 60% on average. For this reason, the analyzed sample showed good quality characteristic as it was the upper limit of the average. It is known that the PLA2 enzyme together with mellitus affect the antimicrobial activity rate of the venom. In generally, PLA2 are considered to be approximately 10 to 12% of the venom. You can see on the table the PLA2 ratio of Anatolian honeybee venom constitutes 20.60% of the venom. From this, it's concluded that PLA2 was higher than the average in Anatolian honeybee venom. In this study, antimicrobial activity of Anatolian honeybee venom sample was tested with both agar-well deletion and mild methods. After incubation period, honeybee venom demonstrated antimicrobial activity against all bacteria and yeast-like fungi. Moreover, with increasing bee venom concentration, the inhibition zone increased and the venom concentration of 50 microgram showed the highest inhibition zone against all microorganisms. The highest effect zone was obtained from Staphylococcus pyogenes, Enterococcus faecalis, and Vibrio sp. The lowest effect zone was against Klebsiella pneumonia subspecies pneumonia. In this table, we can see the mild values obtained using the broad deletion method for different microorganisms varied from 3.06 microgram per milliliter to 50 microgram per milliliter. The lowest MIC values detected to Staphylococcus pyogenes with 3.06 microgram per milliliter. We found that Staphylococcus pyogenes was sensitive to very low concentration of venom. These results show that honeybee venom can be used as an alternative method for superficial infections that are common in patients. You can see on the table that the effect of Anatolian honeybee venom against resistant bacteria such as Staphylococcus aureus, MRSI, is quite high. These results confirm that Anatolian honeybee venom is a candidate for an alternative treatment method against drug-resistant bacteria. It has been reported in previous studies gram-positive bacteria are more sensitive than gram-negative bacteria against bee venom. Vibrio used in this study is a gram-negative bacterium that causes necrotizing fasciitis. The mic values of Anatolian honeybee venom against Vibrio sp was also very low. These results show that Anatolian honeybee venom is also highly effective against gram-negative bacteria, and in cases where these bacteria cause disease, bee venom has an inhibitory effect on the growth of bacteria. The mic value of the venom against Staphylococcus and Escherichia coli were 12.5 microgram per milliliter. The obtained low mic values against Staphylococcus aureus, which causes superinfection in the people with weakened immune systems, show that Anatolian honeybee venom may be used as an effective and natural treatment material for these patients. In conclusion, the analyzed venom show high quality characteristic as it was the upper limit of the mean with a mellitus content of 57.62% and high mellitus mean PLA2 value. Beside the high mellitus content, a broad spectrum of antibacterial, antifungal activity of Anatolian honeybee venom were observed against all tested potentially pathogenic gram-positive, gram-negative, and yeast-like fungi. The active antimicrobial effect of the Anatolian honeybee venom against all microorganisms can be explained by PLA2 content density. It has been suggested that mic values of less than 100 microgram per milliliter are considered very promising in the investigation of compounds isolated from natural source as a new anti-infective agent. When all mic data obtained from this study results are evaluated, Anatolian honeybee venom is a good alternative source of antimicrobial compounds, including resistant microorganisms. Findings from these studies may help design a neat serum or cream containing Anatolian honeybee venom for the successful application in the treatment of skin diseases caused by the microorganism tested. However, he suggested that in vivo studies of toxicity results are still needed before the application is developed. Thank you for your attention and listen to me.
Okay, thank you very much, Dr. Emine Sonmez. A very interesting study. I know the work of Professor Meral Kekecioglu, which I salute with this occasion. I know her myself, and I know the work. I'm waiting maybe collaboration between the researchers from Turkey. With Dr. Emine, with Professor Meral, and Romania, maybe.
Thank you so much, maybe.
If you have some questions about the presentation, please write them in the chat to go on with our schedule. Thank you, Dr. Emine.
Thank you again.
Now I'm inviting Leila Bendi Falah from Algeria to present the paper entitled Daily and Seasonal Variation in the Activity of Wild Bees in Northern Algeria.
Dear Professor Otulia Bobis, before starting the presentation, can you remind the presenters the maximum duration is 10 minutes
Yes, okay. I'm asking all the presenters from our session today, because we have a limited time to speak about 10 minutes for their presentation. Thank you. Dr. Leila, you have the floor.
Thank you. Thank you. I thank you so much for this great event, and I'm happy to be among all the researchers. So, but my selection.
Yeah, please share your screen.
I didn't found my.
You have a red button on the down, which is share screen, and then okay, you started. Share, and now you choose your presentation from there, and it's okay, I think.
Yes. It's okay. Thank you.
It's charging.
So my presentation concerns daily and seasonal variation in the activity of wild bees in northern of Algeria. So plant pollinators and interactions are recognized for their key role in ecosystem functioning and sustainable agriculture. And the mutualistic relationship between plants and pollinators are essential for plant reproduction, ecosystem maintenance, and food security. 75% of the leading global food crops depending partially or fully on pollinators in the world. Animal pollination helps for 35% of global food production, and 80% of wild plants require insect pollination. So major groups of pollinators belong to different groups, and 87.5% of the 352,000 of flowering plants are pollinated by animals. The first group is bees, flies, sunbirds and hummingbirds, moths, and butterflies. The main pollinators in temperate zones are Anthophila, such as honey or managed bees and wild bees, like bumblebees, solitary bees, with overflies as co-pollinators. In the world, we have a superfamily of Apoidae, which has 7 families. We have social bee, like wild bees, bumblebees, and managed bees or honeybee. And we have a second group with wild solitary bees. Of factors of activity of wild bees. which are the main pollinators. So according to several authors, like Wilmer, Gerard, plants and pollinators can respond differently to change in climate. The plant-pollinator relationship.
Excuse me just a moment, Professor Leila. Can you try again, because we don't see your presentation. We just hear your hear you and see your camera. So please try to open again the presentation.
Oh. Mm-hmm.
Yes, now it's okay.
Yes.
Yes.
It's okay.
Yes, please.
So there are agents or agent stressor which have activity on wild bees, which are main pollinators. So for this point, and according to several authors like Wilmer and Berkeley and Gerard et al., plants and pollinators can respond differently to change in climate. The plant-pollinator relationships are particularly vulnerable to spatial, temporal, and morphological. So here we have some agent stressors like habitat and habitat discretion or fragmentation. We have parasites and pathogens like meat, varroa. We have monoculture, climate change, pesticides, and agricultural chemicals. So I want to present you Algerian wild bees and the floral researchers. With its 6 ecological regions, Algeria, the largest country in Africa, has a species rich bee fauna because of its ecological conditions, climate, and diversified flora. According to Dobela and Chetland, floristic richness of 450 taxa exist in Algeria. During the last 2 centuries, several studies on wild bees in Algeria have been carried out. Now, more recently, other studies in Eastern, Central, and Southern Algeria have been carried out by several researchers. And according to Asher and the number of species with bees in Algeria is close to 826 taxa. Now I present the activity of wild bees in northern Algeria. We take into account the appearance of bees and their flowers. The appearance of bees consists with the blooming flowers. So the objective of this work is to show the activity of with bees compared to the honeybee during the flower period in the year 2000. Daily activity.
Sorry, doctor. Again, we see, and all the participants see only the first slide of you.
Okay, I try again. Thank you.
Please, because it's so interesting and nice your presentation. As I see in the right part, it's too bad that we
now is it good? Is it okay?
Try to move the slides.
Okay. Okay. Is it good?
No.
No.
No. stop the share screen and then do again.
Yeah. Okay.
Please. Okay. Now give
Okay.
again share screen. Okay, and put it on full screen.
And now, is it good?
Try to move the slides.
Okay. I move my slides. Yeah. And now, you see?
No,
Ah,
now
la,
it's
ah, la,
oh, it's
la.
disappeared again.
Hmm.
We see all black.
Ah, la, la. Sorry. So I will.
Okay, now we see slide
see?
number 8.
Okay. You see now?
Yes, you can put it on full screen from the bottom, from that.
You can use also F5. If you click on the F5, it is going to be done
Where?
full screen.
Five.
F5 is on your keyboard.
What? I'm sorry. Ah, la, la. And now you see? No.
No.
We see the methodology, maybe.
Methodology, yes? You
Yes.
see methodology? Okay. And are the slides? Yes?
No, it's okay like that if it's working and the slides are going on. You can continue.
Okay, thank you. So for the methodology, the daily activity of the bees, wild bees, and managed bees is analyzed during the months of March, April, May, and June from 6 a.m. The study on seasonal activity of bees took into account the spring of 2000, the year 2000. It's during this period that we witnessed the maximum flowering of natural plants and the significant presence of bees. So here, it's the experimental site of my the National High School of Agronomy in Algiers. Here are the sites of natural plants. We inventoried 65 of natural plants like Docus carota, Andriala, Integrifolia, Fumaria, Agraria, Sinapis arvensis, for example. Here we have some species of wild bees and managed bees. Wild bees, we have social bees like Bombus terrestris africanus. It's endemic species from North Africa. We have also Bombus ruderatus. We have Andrena albopoctata, subspecies funebris, and Eucera notata. So concerning Apis mellifera, we have in Algeria, we have two species of the north of Algeria, subspecies intermissa, and we have the second one from south. So its name is Apis mellifera sariensis. For the result, daily first, I start with the daily activity of natural ecosystem. The days of spring season are characterized by cold and humid mornings. The days of spring season are characterized by cold and humid mornings. So we take in account Apis mellifera, Xylocoppa violacea, and Andrena albopoctata, subspecies funebris. So here we see that Apis mellifera appears the first compared to the wild bees, and because Andrena and Xylocoppa violacea appears when the temperature increase. For the second one, we see average number of Andrena recorded. So in the Andrena number increase in the morning and decrease in the afternoon, because Andrena genus has a short life cycle. For Apis mellifera and Bombus, during April and May, we see that bumblebees is not its number is not very important, because it prefers mountains. And Apis mellifera is very important in the morning. Now, the second objective of result of the secondary objective is seasonal activity of wild and managed bees. Here we see that Apis mellifera is very important because we have hives in the school, and the genus Andrena with two species, Andrena albopoctata and Andrena ferrugineus. Their number is very important compared with Lasioglossum discum and the Megachilidae osmia. Okay. So in conclusion, the results obtained show that the daily activity of the bees studied from March to June reveals that the honeybee flies early. It's followed by wild bees as soon as temperatures rise. Regarding seasonal activity, climatic factors such as temperature and humidity influence bee activity. As such, the abundance of bees studied is positively correlated with temperature and negatively correlated with humidity. This is because Apis mellifera is affected by humidity and slight increase in temperature. Statistical tests show that these two factors have no effect on Andrena flavitis and Lasioglossum discum. On the other hand, the Antophora apericeps, this species, the act favorably while relative humidity doesn't influence on activity of Antiphora atriceps. Therefore, above a certain minimum or maximum temperature and humidity threshold, solitary bees are affected. So thank you for your attention. Here is a new record of wild bees of Algeria, Xylocopa pubescens. So I identified it at the Museum of Natural in Natural Museum in London. And thank you for your attention.
Thank you, Dr. Bendifalah, and sorry to the participants for our technical problem, but I know that Dr. Bendifalah is willing to share the study with those interested if we could not see from the beginning. And if you have questions for this presentation, please put them on chat. Thank you again, Dr. Leila.
Thank you. Thank you. Thank you so much.
We are going further with the other paper. I'm inviting Ulku Zeynep Esertaş from the research group of Professor Sevgi Kolayli, which I salute with respect now, to present a very interesting paper about the antimicrobial, anti-quorum sensing, antibiofilm, and other properties of chestnut ethanolic propolis extract. Please.
We would like to open the voice of this participant, but we couldn't find her name. Please, the participant, please go to the reaction sections and make a sign, and we can open your voice. Now we have found her.
Okay. On page 3, I found Dr. Ulku.
Yes, we can hear her.
Can you hear me?
Yes.
Okay. Hello, dear participants and dear Congress team. Can I share my?
Yes, please.
Okay. Can you see?
Perfect.
Okay. And okay. Dear participants and dear Congress team, hello for everybody from Trabzon. I'm Zeynep Esertaş from Karadeniz Technical University, and I want to talk about for you antimicrobial, anti-quorum sensing, antibiofilm, and anti-swarming activities for activities of ethanolic chestnut propolis extracts. I want to share you with a little video in my video and my bee. And I want to start with Einstein's said, if bees disappeared from the world, it means humanity has 4 years to live. And that seems to be very important for humanity and for the world. So I want to talk about the bees product, propolis. Propolis is very important for us and for healthy, for alternative medicine, for medical, alternative complementary medicines. Propolis is very important for us. And what? Sorry. Why this made this research, this investigate, because you know antibiotic resistance, which has increased rapidly in recent years as a result of uncontrolled, unconscious antibiotic composition, pose a great threat to public health. Now, our big, huge problem is antibiotic resistance. And now, today, in future, maybe 20, 50 years, that's a big problem. Maybe top 3, cancer, antibiotic resistance, and blah, blah, blah. This problem, we have to new treatments, alternative medicines, for complementary medicines, we need new approach. So the indication of the antibiotics has increased the need for new effective and less toxic antibiotic raw materials on antibiotic derivatives. Now, less toxic, that is so important, because you know we don't eat antibiotic, but we eat a propolis. That's a big difference for us. Propolis is a resin mixture that honeybees collect from nature for the hydrogen and safety for their use. The natural bee mixture contains many different polyphenols and oils, has wide biological activity properties. It's frequently used in complementary medicine. Yes, we know propolis is very rich for oils or polyphenols or another things, but we know propolis is so rich and so powerful for our immune system and for healthy, for our maybe activity. So we know the propolis is very important for our body. Now, what we do, how did do? We investigate propolis, antiquarium sensing and antimicrobial activity. How we use determinant by the agar diffusion methods for antimicrobial and antiquarium sensing activity. Now, I want to talk about quorum sensing. This is new approach and a new technique for literature. Quorum sensing is aimed to not kill the bacteria, block the bacteria signal system, and so bacteria alive but not a pathogen. We aim to this because bacteria, like a people, bacteria talk to each other, and so they will be a pathogen. So we dreamed, and researchers think about if we cut the mechanisms, signal mechanisms in communication with bacteria, so maybe bacteria won't be a pathogen. So we don't need an antibiotic. This is the start. So what we do, we start testing our products anti-quorum sensing activity. Which step? Wellison inhibition, anti-swarming activity, anti-biofilm activity. So if our extracts has this activity, this is very important for us for new antibiotic material or new quorum sensing material for long time antibiotic, maybe antibiotic for use long time, because the bacteria not pathogen, so now we won't be ill, so we need to antibiotic, and maybe more strong to war of the antibiotic resistance. This is our thinking, or this is this point aimed. This is basic on our research. So this is our antimicrobial activity result. You can see the our microorganisms and zone diameter, and you can see the control zone diameter and our extracts, chestnut propolis extracts zone diameter, and you can see the similar, especially antifungal activity is similar to antibiotic. this control is amphotericin B, and you can see here, this is antimicrobial activity results, anti-fungal activity results. First picture, 1 day, another picture, second day, and you can see the difference. And our anti-fungal activity results, we wait 2 days. After 2 days, we can see the results and note it. And you can see this one is amphotericin B results, zone diameter, and this is our chestnut propolis extracts disease. And you can see similar, very similar. Yes, you every day you eat, you don't eat antibiotic amphotericin B, but every day you eat a propolis, and little, but you can eat, and that's protect you for antifungal or another microorganisms. Maybe you feel better, and this is important for our immune system and physical activity for routine life. And you can see another microorganisms, Mycobacterium. You can see the zone diameter in propolis, and this is show us the propolis has a very high antimicrobial activity. Yes, you know, that's okay. Maybe you can say, yes, literature is say that. But now I want to say first new. Yes, you can see antiquarium sensing activity, and my favorite bacteria, you can see Chromobacterium violaceum, and it has a pigment, violacin pigment, and it is quorum sensing mechanism because the bacteria grow, so violacin pigment, and so bacteria will be a pathogen. But we cut the violacin pigment, inhibit the violacin pigment, so that's not a pathogen, but live and don't need antibiotics. So that's a new approach, and this is important for the literature for the future. And you can see this is our propolis extracts and zone, and not the bacteria alive. The bacteria alive, bacteria is here, bacteria grow, but not produce violacin pigment. It's cut down to our propolis extracts. That's so important. And we use 2 Chromobacterium isolates, and so our propolis extracts show antiquarium sensing activity are 2 Chromobacterium isolates, and that's very important for us. And you can see the difference antimicrobial activity and quorum sensing activity. You can see here the gentamicin antimicrobial activity, and you can see the antiquarium sensing activity. And same time, this zone is our extracts antimicrobial activity. So our extracts has rich antimicrobial and antiquarium sensing activity. And so this is our 4 studies, and this studies is so important research for. And another step to say 2-step: antiquarium sensing, antiviolacin, antibiofilm, and anti-swarming activity. Anti-swarming activity, we test bacteria, Pseudomonas aerogenes, PAO-1. So this is a typical colony morphology swarming, but we test our extract, change this. Bacteria live grow, but not swarming activity. But our extracts is here not good activity, not anti-swarming activity. We don't see anti-swarming activity, but we see biofilm, antibiofilm activity, but not statistical understand. Maybe future we try again another bacteria we will try, because anti-biofilm activity we only try Pseudomonas, but in the future, in the next study, we try another bacteria. I think our propolis extracts show statistical understand anti-biofilm activity, and so this is very important, because you know biofilm is so big problem for infections, and especially hospital infections is so important. Similar to our study, Surgery and Jaylan determined the propolis extract show antimicrobial and antiquarm sensing activity. That's difference for us. This study not only gram-positive bacteria, but our study show the extracts has antimicrobial activity for gram-negative, and so we say our extracts richer. Maybe it's big. Say, but our extract is richer than maybe another or similar extracts. Our extract is richer than, and we have a potential. We show that our extract has a potential, big potential for this research, and so we need a lot of study, maybe molecular studies, genetic studies, but we need future next step. But in this time, we say our extracts has a potential for antimicrobial activity or antiquarm sensing activity, and quorum sensing activity now very important for antibiotic resistance. My presentation is so enough, and I think I say here, and my lab and my team, you can see. Thank you so much.
Okay, thank you, Dr. Ülkü. It's a very interesting study. Propolis, it's an endless material for medicine and for health, for human health in the future, and we definitely have to be very careful and make as many studies we can, all the people from all over the world, because I think propolis it's future comparable to antibiotics, or to replace antibiotics, because as you said at the beginning, for antibiotics we can have this problem with using our body with them. I have some questions for you, but I will write in the chat, and
okay,
if somebody else have questions for you, I invite them there, and we shall go on with our presentations. Thank you again, Dr. Ülkü.
Thank you so much.
Now it's my pleasure to invite dear Professor Sevgi Kolayli to present another very, very interesting study about the biologically active properties of sunflower honey. Sunflower honey is a very good type of honey, and we also in Romania, our research group have different studies on this type of honey, and I'm waiting to listen. Professor Sevgi Kolayli, please.
Hello, Attila.
Hello.
Nice to see you today.
Yes, it's very nice.
I miss you. Dear guest and dear participant, I don't want your time. Very short my presentation. The name, biological active properties of sunflower honey. In this study, we investigated sunflower honey, some characteristic properties, some physical and bioactive properties. You know, honey is very natural products and sweets, and we use in apitherapy for many purposes, for energy search and for medicinal honey, such as. And our country, you know, is very rich about honeybee products, and sunflower honey, especially produced in Marmara and Aegean region, and this honey is very easily crystallized, and our people, Turkish people especially, doesn't like the crystallized honey, and maybe the honey used as creme honey. And we investigated the honey properties, physical and bioactive components, and our results, you see, total phenolic contents, total flavonoids, total antioxidant capacities, and phenolic compositions, and etc. We studied. Our results showed that the honey is very, very monofloral honey, height sunflower pollen, 80% you see, and the honey physical parameters is similar, light colored blossom honeys, not different the other blossom honeys, but total phenolic contents, total flavonoids, and total antioxidant capacities, not so high, not so lower, a medium degree, but the honey contain especially high amount myrcetin flavonoid and pinosemprin, Caffeicus filnester. It's very interesting results for us, and because the flavonoids is very useful, and today they are used in medicinal purpose, and we say that the honey is very valuable honey, and we look at the literature. Some literature results support our results, such as myrcetin and pinosemprin was found is very high in the honey. It is interesting, these results, and we say that the honey people must use the honey in protect health, but we must use the honey not heated for crystallized, but we must use it as creme honey, crema honey, and our results, that's now. Are there any questions, Tila and another guest? Thank you very much for your nice organization also.
Okay, thank you, my dear professor. If anybody has something to ask, I just want to state that sunflower honey is indeed a very valuable type of honey, but sunflower being an agricultural crop culture, we have to be very careful about the contaminants. We
Yes.
have to,
Yes, you are right.
yes, and
Yes.
neonicotinoids in Romania now we have a big problem because neonicotinoids are allowed in sunflower seeds treatment, and this could be a very big problem.
Yes.
So
Right.
yes, I think that before we use this valuable type of honey, we have to be sure, we have to make analysis, lab analysis, that it is free of any contaminants, because it's a pity for humans, but also for the bees. It could be a problem for the bees also if the culture, if the field of sunflower is treated with some I don't know, insecticides or chemicals.
Yes, you are right, Attila.
Okay, if you have questions, please write them on the chat for Professor Sergi Colaili. I thank you again. We now continue with another friend, which I am saying hello from Romania and hope to meet again. Like Professor Colaili said, I'm giving the floor to Shaban Keskin to present another very good study about propolis. Shaban, please.
Thank you so much, dear Professor Otilia. First of all, I would like to thank all the Congress board members in the body of Professor Ageros to make this event come to existence. I would like to give a presentation about propolis. Separation of phenolic compounds from propolis. We make a new method for the separation, and I will just give the small information about this study. We all know that raw propolis contains many bioactive compounds, and many solvents are used for the extraction of these bioactive compounds from the raw material. And in literature, there are many papers reporting the antioxidant, antimicrobial, anti-cancer activity of propolis as well. And this biological activity is thought to be associated with the phenolic compounds the raw material contains. And main aim of this study is maybe explained in three ways. Our aim is to separation of phenolics from ethanolic extract, and determination of the separated compounds, the biological activity of separated phenolic compounds, and maybe these separated compounds may be used for the preparation of water-soluble products. In literature, I read a paper, Propolis, is there a potential for the development of new drug? And written by Professor Bankova. She is very expert in this area. So in this paper, it's stated that propolis has a potential to find new compounds for drug developments. Some studies also carried out in literature for separation of propolis compounds from the raw material. One is carried out in Croatia. These papers, when these papers are examined, the methods used can be seen in chromatographic methods. Column chromatography, thin layer, or paper chromatography are used in that studies. What we did that we extracted raw propolis with 70% of ethanol, and then we encapsulated the extract with the pectin. Then we used this encapsulated or powdered form of these pectin beads for the separation of phenolic compounds from this pectin propolis beads. So our method is different from the literature. In literature, there are many chromatographic techniques for separation of these phenolic compounds. Now we used encapsulation, firstly encapsulation, and then we separated the phenolic compounds from these microbeads. How we do this? We do it in 2 steps. Firstly, pectin beads were homogenized in water at 1:100 ratio for 1 gram pectin beads in 100 milliliter water. Then these homogenized pectin beads was centrifuged, and the supernatant was collected after centrifugation at 13 RPM for 5 minutes. And secondly, this salt pectin was precipitated from the supernatant by adding the same volume of ethanol, and then the mixture was filtered, and the filtrate was collected and dried in a rotary evaporator. And this dried material contains some phenolic compounds of propolis. And we tested the separation by thin layer chromatography. In the first column here, you see the raw propolis extract, and the second column, the first precipitate, not dissolved part in the water. And the third column, you see the separated phenolic compounds, especially the major ones here. Then we analyzed the chemical composition, and we find that some benzoic acid derivatives, cinnamic acid derivatives, pinosambin, and caffeic acid phenylethyl ester, we get these phenolic compounds in the water-soluble fraction. As conclusion, we obtained 3 major bands by thin layer chromatography, indicating the separation of phenolic compounds. And we obtained completely water-soluble fraction after this technique. And of course, more studies should be carried out for optimization. And maybe these findings may lead to prepare new products containing propolis. Thank you for me giving the chance to make this presentation. If you have any question, I can try to answer.
Okay, thank you very much, Shaban. It's an interesting study. Taking into consideration that water-soluble fraction of propolis could have a very good future, especially in developing some new products for children, for example. So it's not this alcoholic extract involved. If you have questions for Shaban, please write them. And now it's my pleasure to introduce the other part of Shaban family, Keskin family. Love you.
Thank you. Thank you.
And miss you also. And I hope this this craziness in this world will end eventually, and we could meet in person again. So I'm inviting my dear Merve Keskin to present us some other very interesting, very interesting study regarding bee pollen and nanoparticles development. So it's a very interesting study. Please, please, Merve.
Firstly, I would like to say hello to everybody, and I would like to thank to Professor Ulaş and Damla. They are very nice couple, also like us. And I'm very happy to take part in this section because of you, Otilla. You know, I love you very much. Today I would like to try to find present about my new study area. I try to connect the bee products and nanotechnology. So this is my first study. Do you see my presentation? Is it okay?
It's okay.
Yes. So in my first study, I tried to synthesize the bee pollen-based nanoparticles. In this study, firstly, I should work on the nanotechnology. Before, in my PhD thesis, I worked on encapsulation, and in encapsulation, we can obtain the micro capsules. And this time I try to we obtained the micro and macro capsules, and I try to obtain the nano capsules this time. The nano capsules size should be 1 to 100. They should be smaller than the 100. Why I want to synthesize these ones? Because we should this area, nanotechnology area, is and developing area, and we can't connect the nanotechnology drug studies, environment studies, chemical production, or other biological applications. Apitherapy is getting popular, and some of the bee products couldn't use directly, like propolis. So we should do some process to develop new products based the propolis or other bee products. As I said before, nanotechnology or green synthesis of nanoparticles popular in physical electronics. You can see my slides. But we can synthesize the nanoparticles by two methods, chemical and physical methods. If we use the chemical method, using nanoparticles in biological applications are limited, and they are not environment eco-friendly nanoparticles. There is lots of toxic materials in. There could be lots of toxic materials when we try to synthesize by the chemical methods. So we should use the green synthesis technique in this part. Why we choose it? Because there is no toxic material. It's cheap, it's rapid, and fast. We can obtain the particles very fastly. We could use the bacteria, plant extracts, or other biological materials when we try to synthesize by green technique. In this study, I synthesized the silver-based nanoparticles by using pollen. As we know, pollen has many applications and biological activities, and it's rich in phenolic compounds, proteins, and lipids. So I extracted the bee pollen. I prepared a water extract of bee pollen and then mixed it with a solution with silver, and then I solved the other particles. The most important area is this study is the silver reduced to silver zero. So it's a kind of electron transport system. By this system, you can obtain the nanoparticles. After obtaining the nanoparticles, I should characterize them by using a spectrophotometer, FTR, and scanning electron microscopy, because as I said before, nanoparticle size are important for us. The maximum absorbance was at 3048. It's correlated with the literature. And functional groups, the BP with red one, the proper pollen extracts FTR data, and BP silver nanoparticles with the green one. When we see the functional groups, we sure that the functional groups is active and they are take part the reducing of the silver. And then we take the scanning electron microscopy pictures, and we saw the nanoparticles has an average of 50 nanometers. They are changing 40 to 60 nanometers. And after that, I try to find out the biological activity properties of the nanoparticles. For this purpose, I work on enzyme inhibition properties, because in diabetes mellitus, alpha amylase inhibition and alpha glucosidase inhibition is important. So I first characterize the bee pollen extract. I try to find out the total phenolic content and also nanoparticles total phenolic content. And then I perform the damp on the enzymes, and I find that they are effective, very effective on the enzymes. So this is my last newborn baby, this article. We can say that the nanoparticles are very effective on the many enzymes. I'm trying to synthesize the propolis based nanoparticles also. We can change the nanoparticles metallic ion. Also we can use the gold, we can use the silver. So it's a new and important area for the apitherapy applications. I know we don't have any time. I should be very fast. I would like to thank to all participants. Thank you very much for your attention.
Okay, thank you, dear Merve. In the end, your presentation was not working, or maybe it was finished. But I imagine that all these presentations will be available for anyone which is interested.
Yes, I can send by email.
Okay, thank you, my dear.
You're welcome.
And now I have to make a change in our written program, because we have two colleagues from Ukraine, which I want to invite them present their papers. We all know that there the situation is not so pink, unfortunately, and the connection, the internet connection may fall. And please, I want to ask you to allow them to present their papers in the following minutes. And now I want to ask our colleague Irina Kuz from Ukraine to present the paper Oral Hygiene and Periodontal Indices in Children with Type 1 Diabetes mellitus during gingivitis treatment. Please, Irina.
Do you hear me?
Yes.
Good morning.
Good morning.
Thank you for understanding. Do you see my presentation?
Now you start sharing the screen.
I shared it.
Okay. We'll wait a little.
Do
Yes.
you see it?
Yes, it's perfect. Please.
I'll try to change my slides. Are they changing? Yeah.
Yes. Yes.
Mm-hmm. I'll begin and I shall do it quickly. Dear head of the section, dear experts, dear colleagues, we present our research work, oral hygiene and periodontal indices in children with insulin-dependent diabetes mellitus, gingivitis treatments. The presence of first-type diabetes mellitus in children is the main risk factor for the occurrence of inflammatory processes in periodontal. It leads to the progression of inflammatory and degenerative processes in the oral cavity. That is why it is very important to establish the etiological factors and pathogenetic mechanisms of gingivitis in children and create new methods for prevention and treatment of this dental disease. The aim of the study was to assess the state of periodontium and to evaluate the quality of oral hygiene in children of primary school age with type 1 diabetes mellitus under the influence of treating scheme of chronic catarrhal gingivitis. We determined the oral hygiene indexes according to Fedorov-Volotkina and a simplified oral hygiene index according to Green-Vermeulen. And also we studied PMA index in Parma modification, the bleeding index according to the law and sealnets, which is also called gingival index. And we determined the bleeding index according to Müllermann. We examined 82 children from 6 to 12 years old. 56 of them had type 1 diabetes mellitus and 26 children were healthy somatically. We divided them into such groups. First one was children with healthy periodontium and no concomitant diseases. The second group was children without concomitant diseases, but they had chronic catarrhal gingivitis. The third group complaint of children with type 1 diabetes mellitus without periodontal inflammation. And the first group consisted with 1 diabetes mellitus and chronic catarrhal gingivitis. On the next slide, I present my certificate about created treating scheme of chronic catarrhal gingivitis in children with first-type diabetes mellitus. We taught all the children how to perform oral hygiene in the right way. We asked them to use soft toothbrushes and toothpaste with extracts of grape. We treated chronic catarrhal gingivitis in second group according to the standard protocols of Health Ministry of Ukraine. And we did chronic catarrhal gingivitis in the children with diabetes mellitus with next scheme. Our scheme included the antioxidant ascorutine with routine, and then we asked them to rinse the oral cavity with antiseptic solution Sengiva 2 times a day, and to absorb the probiotic Biohaya Prodentis. There is such a probiotic like gum in Europe. special probiotics for oral cavity. And our treatment scheme lasts for 1 month. We carried out the evaluation of results in 1 month, in 3, 6, and 12 months. The next slide demonstrates oral hygiene in the first group of automatically healthy children. Then there is information about satisfactory oral hygiene and mild inflammation in somatically healthy children with chronic lateral gingivitis before treatment. The next slide, we see gingival indices in children with type 1 diabetic mellitus and healthy gums. They were equal to 0, and the oral hygiene was also satisfactory. The last table is the most noteworthy. We would like to discuss this data more detail. There we can see 2 photos which illustrate mild inflammation in somatically healthy children with chronic lateral gingivitis and moderate inflammation in children with type 1 diabetic mellitus. The oral hygiene indices, according to Fedorov Volodkina and Green-Vermeulen before treatment, showed us the unsatisfactory level of oral hygiene. The oral hygiene of these children improved to a good level after 1 month. Then the level of oral hygiene stabilized and continued to remain at the same level after 3 and 6 months. These indices slightly increased and corresponded to a satisfactory oral hygiene after 1 year. They were respectively 1.26 and 0.47 times less than they were before treatment. The PMA index in the group of children with chronic lateral gingivitis, type 1 diabetic mellitus, was 40%. It's rather high. It indicated a moderate degree of inflammation process in their periodontium. This value decreased more than 4 times and amounted to 9%. This is a very mild inflammation. And above index slightly increased, but was on the average 2 times lower than before treatment after 3, 6, and 1 year, 3, 6 months and 1 year. Gingival index according to lower illness and bleeding index according to Mulliman before treatment also indicated the average degree of inflammation after 1 month. These indices decreased to level of mild inflammation. These indexes stabilized at the level of mild inflammation after 3 months, 6 months, and 1 year. In 1 year, we have the following results. PMA became 2 times less. Gingival index, according to lower decreased in 1.5 times. Bleeding index in 1.7 times decreased. They became lower than before treatment. In our opinion, we should pay great attention to choosing medicaments. First of all, professional hygiene and correct oral hygiene at home is important. We also insist on prescribing antiseptic solutions and, of course, oral probiotics for further normalization of oral microbiota. Using medications with antioxidant capacity like ascorutine, here is rutin and vitamin C in it, is significant in children with chronic lateral gingivitis with type 1 diabetic mellitus. Thank you for your attention.
Okay, thank you, dear Irina. Congratulations. I felt that you have a little afraid, but it was perfect.
Unfortunately, we have such a situation in our country.
Yes,
I'm
yes.
very. I'm not at home now. I'm in a village.
Oh.
Thank you.
Let's hope and pray that everything will be good, and then people will have something in their heads, because
we run away from our city.
that it's terrible. It was anyway. It was an interesting study. Diabetes is a disease that many people have, many children have, and all the studies that are made in this direction, they are very welcome. And I wish you and your colleagues to be able and to carry on these researchers in good and safe environment. Thank you again, dear Irina.
Thank you very much.
And now let me present you our second colleague from Ukraine, our colleague from animal science, Dmitro Kristenko, which will present us a paper about the legislation for sturgeon bycatch during commercial fishing.
Okay, hello,
Please.
can you hear me?
Yes, we can hear you.
Okay, so thank you very much for the word and thank you for understanding. Situation is really difficult, but we all understand. Okay, I will share my presentation. Okay, can you hear it? Can you see it?
Yes, yes, we can.
Okay.
Thank you.
Okay, so this study was made under cover of the World Wildlife Fund of Ukraine. And actually the main issue was that sturgeons are highly protected red-lead species in Ukraine, but probably in the main countries of our world. And the issue is that sometimes they appear in commercial catches as bycatch. And first, the specialists from WWF, and then we specialists from the National University of Wildlife and Environmental Scientists and the Institute of Fisheries of Ukraine started thinking about the situation why commercial fisheries, so in legal commercial fisheries, there are no reports within the last 3 years about any bycatch of sturgeons, while in catches of poachers, they are present and now the number is increasing. So, when we started investigating that, we realized that there are two levels that protect sturgeons. So first one is international level. So there are European red list, the Conservation of International Trade and Endangered Species that commonly called CITES. There are Berne Convention, Bonn Convention with all different attachments. And Ukraine took responsibility to protect sturgeons by all those international level documents. However, the issue is that national legislation is developed only based on the Red Book of Ukraine. So in Ukraine, we have a special law stating that, that there is a Red Book that is the list of endangered species that must be highly protected. Unfortunately, it doesn't say anything. So other Ukrainian legislation doesn't say anything about all these international documents. There are commercial fishing rules that also stated that there's species. It doesn't say sturgeons. It says just red list species or species from the Red Book. And the same is, for instance, for commercial species and their resolution for compensation for illegal harvesting of these species. This is a very huge bottleneck because while governmental authorities don't foresee this as a problem because we, Ukraine, fulfill all international responsibilities regarding sturgeons, but if there is a small chance that sturgeons can be dropped out of Red Book of Ukraine, it will be a very interesting, very strange situation because in this occasion, so nothing will protect them more. Why this can happen? Because sturgeons are not only red list species, they are valuable objects for aquaculture. And therefore, some commercial enterprises would like to facilitate the work with them. The matter of fact is that procedure of getting legal permits, so we can get permits for harvesting sturgeons for scientific purposes and for breeding purposes. But the procedure is very unclear, and some territorial bodies of Ministry of Environment that must authorize requests does not exist anymore due to reform that had happened. Therefore, in practice, I tried twice in 2020 and 2021, and I failed. I cannot get the permit to harvest sturgeons for commercial purposes. The same with for instance, state-owned Kherson sturgeon fish plant, who is this public body that must stock sturgeons into the Black Sea, and so on and so forth. Therefore, when we try to understand what is the manual for sturgeon bycatch, so we realize that according to Red Book, species should be prohibited from removing from the wild. They're banned to remove. In commercial fishing rules, there is a very interesting story, because it said that alive sturgeons in bycatches should be released, while the dormant fish and other aquatic living resources must be accounted by the egg and recorded in catch and effort logbook. That is very strange, and this is commercial fishing rule. This is a rule that a commercial fishers organization must obey. Therefore, we have some temporary document that calls Black Sea Fisheries Regime. It's renewed annually every year. And here it says that it should clarify different unclear positions. So in this document, we said that sturgeons in bycatches must be returned to the habitat in any form. So, but unfortunately, as far as I know, nowadays we have also this high sturgeon of reform, and this norm was removed from the regime. So situation nowadays in 2022 appeared to be very funny. So if commercial fishery enterprise, according to commercial fishery rules, will account dormant sturgeon, they automatically violate other part of commercial fishery rules, that they do not exceed the volume of allocated quotas. So for this violation, they will have a fine about from 11 to 14 euro plus compensation for illegal harvest. And this is from 16 to 3,500 of euro. That is very painful. I would like to emphasize that this is a punishment that will occur to commercial fishery organizations if they will act according to procedures prescribed in commercial fisheries rules. While if they violate this and just drop sturgeons in any stage in the water, their fine will be from 11 to 14 euro, and that's all, because in this occasion there will be no compensation for illegal harvest. And even more, the proving of users' guilt if they just drop sturgeon back to the water that violate commercial fishery rules will be very challenging, because, for instance, the user can deny that they didn't harvest at all because there is no sturgeons on board. And if there are some records or some tapes or some photos, they can claim that in time of removing sturgeon was alive. So this is a very interesting situation, while in legal plan of Ukraine, when the punishment for following the rule is higher than for violating it. So for this, we propose do some changes for the legislation. And actually, we raise another question, that beforehand, we have a normal situation that the sturgeon must be just reported and dropped back to the environment. But for the last 3 years, so State Fisheries Agency territorial bodies did not report any bycatch from commercial fisheries, while in poaching catch, fish catch, they are just rising. Then the question arises, how does it happen? So we just made our small inside investigation, and actually we understood that commercial fisheries organizations don't want to report bycatches because of the reaction of control bodies. Because, for instance, ecological inspection and fisheries protection authorities, when they notified about bycatches, they started increasing the control in certain area. And if they found 3 or more bycatches cases, so just cases, they can prescribe users to change the commercial fisheries area or close the commercial fisheries area at all. This automatically will lead to that the commercial fishery organization will not fulfill the quotas, and then they will have troubles at the end of the year. Therefore, commercial fishery organizations are not in favor of appropriate reporting. When we started investigation further, we realized that, first of all, we proposed that we should remove from our legislation any possibility for punishment for bycatch reporting, because this is really weird. We should understand what the situation is, what species are, how they are sampled, so how much are they. So secondly, the reporting mechanism should be developed. So it should be clearly stated. So in what form, what data should be provided. So it should be some Telegram channel or something else, or fax or teletype or whatever they use, we don't know. And third, it should be necessarily prescribe the position, so real position for closure of commercial fisheries areas. Because nowadays we have a situation that limitations that we have simply doesn't work. And so Institute of Fisheries counted, and they said that the red list species should not exceed 5 to 10% in bycatches. So my colleague from WWF US argued that it's too much, but unfortunately the situation now doesn't work, and we must understand the real situation and the real picture, and then we will understand what to do and how to proceed with all this. Because nowadays we have something that you see on this picture. This picture was taken by my colleague from WWF Romania. This is Russian sturgeon and sterlet on the farmers market in Odessa. So these species are just freely sold in farmers markets. So what do we propose second? So as the second poll, we propose something like court reform. So all users must understand that they cannot overcome penalty for law violation. Because nowadays in Ukraine, this crime does not recognized as very hard. So it can be delayed. And after 3 years of delay, it can be closed because it exceeded the term. And another thing that we propose is fight against corruption, because Ukraine, in my imagination, before today's, it was like not a developing country, it was a country in transition. So I cannot say that our control bodies are lower educated, so they actually know what are they doing. They're very good and very nice professionals. So they must be nicely paid, and they must understand that for violation of a law, the penalty will be real, but not like it now. So I would like to thank you. I shortened my presentation because I understand the time. So if you have any questions, feel free to ask. Thank you very much.
Thank you, Dmitro. If you have questions, please put them on the chat, and I'm sure that our colleague will answer gladly to you. And now we continue. I apologize again for the changing in the program, but we are understandable with our colleagues from Ukraine. In the program now, it came a very, very interesting presentation. I am asking Dr. Yusuf Zengin to share, yes, thank you, to share the screen. I know that my colleagues from the department will be watching you, because beside the beekeeping practices and bee products, our department is also interested in silkworm rearing. Please, Dr. Zengin, you can start.
Thank you, dear President. Can you hear me? Yes. Firstly, I am so sorry about war. I will pray all colleagues and all Ukrainian people. Inshallah, the world soon the end. Hello, dear scientists. I am happy to see again all of you. I work as an agriculture engineer in International Center for Livestock Research and Training. Before starting my presentation, I would like to thank you, especially dear Professor Ajaroz and him team. Also, thank you all participants. Let's make a start. My presentation is about our project, which supports General Director of Agricultural Research and Policy. My presentation titled The Effects of Differences in Feeding Numbers on some growth and cocoon features in commercial hybrid silkworms. This is my team also. Table of contents I will share with you. Introduction, the aim of study, materials and methods, results, image of project. Let's make a start with introduction. Making has need many things for living science. Its extent, clothing is one of these needs. Silkworm breeding in Anatolia starts about 1,500 years ago and continues today with a minor extent. Also, silkworm breeding and agriculture activity is very important because it supports rural development, does not require too much investment and offers sale, which is a value-added product of economy. In addition, also, silkworm is an important gene source. My presentation aim in this study the effects of different feeding numbers 6, 4, and 2 times a day on the growth and some cocoon parameters of silkworm larvae were investigated in the condition of the institute, which is International Center for Livestock Research and Training. Let's continue. Materials and methods. The material of study is commercial hybrid silkworm. We get the Kozabirlik in Bursa. The study was designed with 3 groups and 3 replication for each group. The height and weight of 25 silkworms larva samples for each repetition were measured before the first feeding at the beginning of each age. In addition, we get some cocoon parameters. These are weight of 100 cocoons, number and weight of cocoons per liter, weight of 40 cocoons with chloride and without chlorides. Also, thickness, lengths, and diameters of the cocoons were determined after cocoon removal. Let's continue the results. First table shows larva weights by age. We totally get average 25 larvas. Larva weights increase more in the group feed 6 times than in other groups for each age. Let's continue the second table. Larva length, it's about. Also, we observed the group 6 feet fed, the better length development. Table 3 shows time difference in starting cocoon weaving. Totally days, I think its importance value because the days, more work, and the other things. 6 feet group, 6 groups start to spin cocoons at 28 days, but 2 feeding groups start to secrete at 35 days. The other table is 4. You see the cocoon lengths and diameters. Also, 6 feet more higher the other groups for these values. Let's continue table 5. It's about some cocon, the other cocon parameters. I believe the most important value in this table, silk wealth. We see the silk region is highest in 6 feeding group. I continue some sharing image for project. Let's start forward preparations. Also the others. Bring the materials to Institute from Kozabilik Institute, the other photos. Also our Institute, Mulberry Garden. Analysis of leaves. First age, weight and height measurement. For second age, you see the larva. Third age also. Fourth age. Fifth age. Leaf harvest, you see the photo. Lime application and bottom cleaning, you see. And another photos, suspender placement. And also finally cocon harvest and cocon analyzers. Thank you so much to listen.
Okay, thank you, Dr. Yusuf, for this this nice presentation. I recognize some of the pictures from our department, from our mulberry garden in the university, from the places where our silk worm are grown by my colleagues. And I have to say that between 2 feeds and 6 feeds, there is a great difference, because a high amount of work and leaves to be collected. If you are growing, I don't know, several beds with I don't know, the 5 or 6 levels, it is very difficult to feed 6 times,
yeah.
6 times the silkworm.
I work full time in a day to get leaves and to feeding larvae. Also, I am happy to interest our presentation photos. President, thank you so much for this.
Okay, if somebody have questions, please put them in the chat so we can go on. Thank you again, Dr. Yusuf. And now I'm inviting Boran Karatash for his presentation, research on determination of optimum ratio of PCR components in gene expression studies. A very interesting study again, and I know that my colleagues will be also interested in this.
My voice come?
Yes, we can see the screen.
Hi everyone. Our study research on determination of optimum reserves of PCR component in gene expression studies. qPCR is widely used PCR method that provides simultaneous observation of nucleic acid amplification. It is also indispensable for gene expression studies. Gene expression is used for the transformation of genes in the form of DNA strands into functional protein structures. The most common measure of gene expression is concentration. It is usually performed to compare the mRNA level of gene in different cells. For quantitative PCR in gene expression studies, generally right and left primer or forward and reverse primer, PCR master mix, cDNA, and nucleus-free water and mix are prepared. However, they put a lot of effort into the optimization the components used to determine gene expression in real-time PCR studies. The trial and error method is mostly used when determining the component and amount of the mixture used in the PCR process. Since this situation continues until suitable rays are found, it causes waste of consumables, labor, and time. In addition, although the PCR process is repeated with the mixture components, the desired results are not always achieved. This situation may cause the trial setup of the study to be established because the amount of cDNA obtained from living material decreases in each PCR process. In this study, it was tried to determine the Optimum ratios of the PCR components for gene expression studies. In the study, RNAs isolated from rainbow throat larvae were used. The larvae were placed in sterile ethanol tube and 0.25 milliliters of RNA later was added to the cup and dried to the aquaculture department laboratory. The sample were sorted minus 20 degrees until the RNA isolation process. Total RNA isolation in larval sample was done with drizzle. The absorbance ratios of the obtained RNAs were examined at the 230, 260, 280 nanometer in the nanodrop device. The cDNA library was created on the same day using the cDNA kit with the obtained RNAs. High capacity cDNA reverse transcription kit was used to generate cDNA library from total RNA samples obtained from rainbow throat larvae. The kit was used according to manufacturer's instructions. The absorbance ratios of the obtained cDNA were checked at the 130, 260, 280 nanometer in the nanodrop device. Obtained cDNA were sorted minus 20 degrees until quantitative PCR. In the study, rainbow throat growth hormone gene and 18S RNA gene region, which is the rainbow throat identity gene, were used as a reference gene. Primer designed by Karataş were used in the study. In the study, the amount of the components were changed and different PCR processes were performed in time until the appropriate ratio was found. The final volume was initially determined as 20 microliters. At this stage, the primer temperature specified by the company synthesizing the primers was taken into account in order to determine the analytic temperature recorded in the PCR process. The temperature of the primers of the growth hormone gene and the primer of the 18S RNA gene were average. In addition, a trial was conducted with 1 degree of the average. Then, if this temperature manipulation were carried out by changing the amounts of right and left primers, PCR master mix, cDNA, and nucleus-free water with total volume of 20 microliters. In addition, nucleus-free water was used on some of the components mixture and not with some. Total volume of each difference ratio used reduced the cost and determined the final volume was reduced from the 20 microliter to 10 microliter and then 5 microliter respectively. Some of negative results obtained at different rates in the figure can be seen. When the ratio of 4:1 was used in the study, the result in the figure was obtained. To reduce the cost and determine the final PCR kit volume was reduced from 20 microliter to 10 microliter and then 5 microliter respectively. The negative result in the figure was obtained when the volume was 10 microliter. The negative result in the figure was obtained when the volume was 5 microliter. After it was understood that the ratio of 4:1 was appropriate, many trials were made and the results in the figure were obtained. Successful results were obtained with this ratio applied each time. As a result, editing nuclear-free water to the PCR component in the study caused the data not to be taken. With the appropriate and many temperature optimization, it has been understood that the ratio of the 4:1 is appropriate and the final volume in the PCR tube should not be reduced below 20 microliter. Thank you very much.
Okay, thank you, Dr. Boran Karataş. We have some problems with the sound, but I believe that everyone who was interested to learn more about your method can ask you directly, and you will share with everyone who's interested your findings. Thank you again for your presentation. And now we go on with another interesting study presented to us by Esen Polat, something with bacteria and a very long and I believe interesting study. Okay, Esen, you may share your screen and we are waiting to hear your presentation.
Geri gelmiyor. Bir dakika, bir saniye. Yalnız başta değil. Hello everybody, I am Untitled Polat. I am working as veterinarian at Central Fisheries Research Institute in Turkey. I am going to present the study titled Lactococcus garvia isolation from vaccinated and non-vaccinated cultured fish present in the same aquatic ecosystem and the antibiotic resistance of the isolated bacteria. In recent years, aquaculture activities have accelerated in Turkey in suitable sea and freshwater environments. The development of fish production brings about fish diseases and, as a result, unexpected economic losses. It is a causative agent of Lactococcus and has been isolated from a wide variety of animals, including cow, buffalo, dog, cat, and fish. Lactococcus garvia, the bacterial pathogen causing Lactococcus, was first isolated from a yellowtail in Japan and has afterward been found in many areas where salmonids are cultured, such as Turkey. This bacteria causes large economic losses in the aquaculture environment. Lactococcus garvia is gram-positive, facultative anaerobic, non-motile, non-spore, oxidase and catalase negative, and zoonotic bacteria. The sampling was carried out in 4 group fish cats in the study, which are vaccinated group, non-vaccinated group, moribund fish and dead fish. In microbiological diagnosis, the head, kidney, spleen, and liver samples of fish were aseptically streaked on tryptic soy agar and incubated at 13 Celsius degrees for 24 hours under aerobic conditions. The isolates were subcultured on the same media to check the purity of the isolate. The pure colonies were typically characterized by gram staining and the following biochemical tests, which are cytochrome oxidase, catalase, and motility. In molecular diagnosis, bacterial DNA was extracted by using the KIAMP DNA mini kit. The identification of the bacterial colonies was performed by PCR assay. As you can see in the image, we used two primers for PCR assay, a forward primer and a reverse primer, which were designed by Zlotkin et al., were used for PCR amplification of the small subunit 16 as ribosomal RNA gene sequence of Lactococcus garvia. In the left image, you see the macroscopic image of Lactococcus garvia. As you can see, gram-positive single or short chain cocci. In the right image, you see the PCR image of Lactococcus garvia. As you can see, PCR samples give the same radiation at 1100 BP as the control sample, confirming that all samples are Lactococcus garvia strains. As a result, Lactococcus garvia were isolated from 4 out of 30 fish in the vaccinated group, 10 out of 30 fish in the non-vaccinated group, 16 out of 22 fish in the moribund group, and 21 out of 35 fish in the dead group. Other bacteria are unidentified gram-negative bacteria. The antibiotic susceptibility of Lactococcus garvia and other isolated fish pathogenic bacteria were determined by the disk diffusion method as described by the Clinical and Laboratory Standards Institute. As you can see in the right of the slide, 10 types of antibiotic resistance were examined. According to results, Lactococcus garvia strains are resistant to sulforamycin, oxalic acid, and gentamicin, but sensitive to penicillin, doxycycline, erythromycin, oxycycline, fluorphenicol, amphloxacin, and amoxicillin. As you can see, other bacteria strains are the most resistant to penicillin and the most sensitive to amoxicillin. As a result, inactivated vaccines against Lactococcus have been used in this aquaculture activities area. The study results show that the vaccines protect the cultured fish against Lactococcus. Thank you for your attention.
Okay, thank you very much, Dr. Esen Polat, for your presentation. If somebody have something to ask, I know fish rearing is a very important section of animal science, and we have to be careful because more and more the water is more polluted, more and more the fish develop different diseases, and this study from aquaculture is very, very, very important. Thank you again. Okay, and we have to go on now. I am inviting Dr. Maciej Kochanowski to present their studies regarding proteomic profiling and functional characterization of the secretome of Anisakis simplex L3 larvae.
Good afternoon. Thank you for the opportunity to participate in the Congress. My presentation describes a study on the proteomic profiling and functional characterization of the secretome of Anisakis simplex third stage larva. Anisakis simplex is important foodborne nematode which can cause a human disease called anisakiasis. This parasite has indirect life cycle. Marine mammals are definitive hosts. Fish and cephalopods are intermediate or paratenic hosts, while crustaceans are intermediate hosts. Humans, which are accidental hosts of Anisakis simplex, are infected by third stage larvae, the source of which are infected marine fish or cephalopods. Anisakis simplex is mainly distributed in the northern Atlantic and Pacific Oceans. These features show the morphology of the third stage larvae of Anisakis. The larvae are wide and about 2 centimeters in size. Anisakiasis is parasitic disease that affects mainly gastrointestinal tract. And in such cases, patients typically have symptoms such as abdominal pain and vomiting. An important form of the disease is gastroallergic anisakiasis, in which abdominal symptoms are accompanied by allergic reactions such as urticaria, angioedema, or even anaphylaxis. The secretome proteins of Anisakis simplex and other nematodes belonging to the Anisakidae family have not been explored. Despite excretory secretory proteins play a key role in pathogenicity and host-parasite interactions. Therefore, the aim of this study was to conduct proteomic profiling of Anisakis simplex third stage larva secretome. The workflow of the study was as follows. Firstly, larvae were collected and identified. Next, 3 batches of secretome proteins were prepared. Then secretome proteins were analyzed using SDS page. After electrophoresis analysis, protein samples were processed and subjected to LC-MS, and subsequently identified proteins were analyzing using different bioinformatic tools. During sample processing, samples were subjected to standard in-solution digestion, and samples were measured using Q-exactive mass spectrum method coupled with nano HPLC. 158 proteins that occurred in all 3 biological replicates were detected. These proteins were classified into 143 protein families. The most frequently represented family was immunoglobulin-like fold. 21 proteins were classified into a conventional secretory pathway, and 77 proteins were assigned to unconventional protein secretion pathway. Gene ontology annotation and enrichment analysis were performed also. A large variety of geo terms was detected, which is quite typical for the secretome of nematodes. The most abundant and most enriched geoterms were, in general, these related to the glycolytic processes larval development, antioxidants, and cuticle. And these annotations cover functions that are important to parasite metabolism, lifestyle, and survival. 72 proteins were assigned to 6 enzyme classes. The most abundant classes of enzymes was hydrolases, and the less represented were isomerases, transferases, oxidoreductases, translocases, and lyses. No proteins belonging to the ligase class were detected. Furthermore, 38 enzymes were overrepresented in the secretome compared to the whole proteome. The KEK pathway profiling revealed proteins involved in 44 pathways. Among them, the most frequent was KEK metabolic pathway, while the main enriched KEK pathways is related to carbohydrate metabolism. Using 3 different bioinformatic databases, we identify 9 potential pathogenicity-related proteins. The majority of them are heat shock proteins. Of all identified proteins, 1, Anis4, is listed by World Health Organization and International Union of Immunological Societies. Furthermore, 18 potential allergens were identified using bioinformatic tools. Most of potential allergens were homologs of nematode and arthropod allergens. The protein interaction networks was established using string server. 53 proteins involved in the interaction network were revealed with high confidence, high prediction confidence. The majority of these proteins were associated with KEK metabolic pathways, particularly carbon metabolism. 18 protein of anisakis and 87 proteins, human proteins, were identified in the host-parasite interaction network. Among human proteins, polyubiquitin showed potential interactions with the highest number of anisakis proteins. 5 proteins of anisakis secretome and 19 proteins of Atlantic herring were predicted in fish parasite interaction network. All anisakis proteins identified in the interactome were identified also in human parasite interaction network. The most highly represented fish proteins involved in the interactome was RAS superfamily proteins. In conclusion, this study represents first global analysis of anisakis excretory-secretory proteins. Detected proteins play an important role in many biological processes and provide a better understanding of anisakis simplex survival, development, and invasion strategy. The identified secretome proteins could be used as a potential targets for new drugs, vaccines, and diagnostic assays. And future in vivo and in vitro studies are needed to confirm our findings regarding the role of detected proteins. Our study was recently, a few days ago, published in the Pathogens, so all details you can find in our paper, and thank you for your attention.
Okay, Dr. Kochanowski, thank you very much, and congratulations for your presentation and for your paper. Also, we know all how hard it is to publish nowadays, because more and more researchers exist, and it's more difficult to publish a paper, especially in ICI indexed journals. Congratulations. If anybody have some questions, please write them in the chat, and I'm sure that Dr. Kochanowski will answer you. Thank you again. Now we are going ahead with the study that Nimra Mubin will present us again another study from aquaculture. Please, Dr. Nimra Mubin, share with us your...
Dear Professor Otila, she is not available now here.
No.
I am trying to contact her. If you say okay, we can continue with the next presentation.
Yes, if then if Hatice Pelin Aslim is yes, I see that she's available. Hello, dear Pelin. We invite you to present your study.
Okay.
Okay, yes, we see you. Good luck.
Thank you, dear lecturers. Thank you, Chairman, dear lecturers and colleagues. I am going to talk about in vitro antiviral activity of neem bark extract against bovine coronavirus, bovine herpes virus 1, bovine parainfluenza virus 3, and bovine enterovirus. Neem is a native plant from neem tree. Other names are Azadibric, Indic, and Indian lilac. Neem leaf and its components were reported to exhibit immunomodulatory, anti-inflammatory, anti-hyperglycemic, anti-ulcer, antifungal, antibacterial, antiviral, antioxidant, antimutagenic, and anticarcinogenic properties. Coronaviruses cause different clinical syndromes such as respiratory tract infections, enteritis, liver, neurological disorders, and nephritis in a wide variety of species. Coronaviruses in the family Coronaviridae are positive stranded viruses with the largest genome are among RNA viruses. Bovine coronaviruses belong to group 2 and are pneumoenteric viruses that cause neonatal cough, diarrhea, winter dysentery, and respiratory disease in cattle. Bovine herpes virus 1 is one of the 8 herpes viruses isolated from naturally infected cattle. BHV1 belongs to genus Varicella virus of subfamily Alpha herpes virini of the family Herpes viridae. BHV is mainly associated with 3 main clinics, IBR, IPV, and IBB. The virus can also cause a wide variety of syndromes such as abortion, infertility, conjunctivitis, and encephalitis. Bovine parainfluenza virus type 3 is an enveloped, non-segmented, negative stranded RNA virus in the respirovirus genus. The clinical presentation of BPIV3 infections can vary significantly from asymptomatic to severe respiratory disease. Disease-related cases of BPIV3 are usually characterized by mild clinical manifestations such as cough, fever, and runny nose. Bovine enteroviruses are small, non-enveloped, single-stranded, positive-stranded RNA viruses belonging to genus Enterovirus in family Picornaviridae. BEVs isolated from cattle worldwide have a wide variety of clinical manifestations, including abortion, stillbirth, infertility, neonatal deaths, as well as diarrhea and fever, dehydration, and weight loss. In the study, the anti-BCV, anti-BHV, 1 and anti-BPIV3 and anti-BHB effects of neem bark extract on target cells were investigated. It is thought that this information will open new avenues for use of NBE as a potential microbicide. The plant extract to be used in the study was commercially available. Antiviral activity studies, antiviral activity of neem bark extract, determination of TCID50 of virus-infected cells, and change in TCID50 values when infected cells were treated with neem bark extract was evaluated. In this test, separate 96 well plates were used for each virus. The antiviral activity study was studied separately for each virus species to determine the antiviral activity on both replication and adsorption. Effects of neem bark extract on virus replication. The wells in the plate were numbered up to 10 from top to bottom and were divided into 2 wells with and without antiviral agents. The last 2 rows of cell and virus on plate surface were used as a control group. 100 microliter of growth medium containing non-toxic maximum concentration of neem solution was placed in the sections that will contain neem extract 4 columns and 4 rows up to 10. Likewise, 100 microliter of cell growth medium that didn't contain antiviral was added to the vase in the other half of plate for the cells to metabolize the neem extract. The plates were incubated for 2 hours at 37 C in a 5% carbon dioxide incubator. Close the end of the incubation period, virus solution were diluted serially between 1 to 10, 1 to -1 and 1 to -10 according to log 10 base. Virus suspensions deleted with 100 microliter in each well were transferred to the wells that were treated with neem extract and did not contain neem until the 10th deletion step. Virus deletions which were left to incubate with cells in an incubator with 5% carbon dioxide at 37 C for 1 hour were removed from the wells at the end of the period and the wells were washed with PBS. Growth medium containing the highest concentrations of non-toxic neem extract was added to wells that were previously incubated with neem. 100 microliter of growth medium and 100 microliter of neem-free growth medium to wells in the other half of plate that was not treated with neem extract, and plates were transferred to 37 C incubator with 5% carbon dioxide. The cells inoculated with the virus were checked daily with a tissue culture microscope for pathogenic effect of virus on cell, and the test was terminated at the end of 72 hours to determine the infective dose of viruses in wells with and without neem. A 50% endpoint titer of virus was calculated according to the method. The effect of neem bark extract on virus attachment. The plate design was designed as in replication study. The cell suspension was transferred to 96 wells placed at 90% monolayer and incubated for 24 hours at 37 C in a 5% carbon dioxide incubator. At the end of the period, the virus groups in which the antiviral activity was to be studied were serially diluted with a non-toxic maximum concentration of neem solution in the range of 1 to 10 to 1 to 10 to -10 according to log 10 base. Simultaneously, reconstitutions of virus with a cell generation medium containing no substance was performed separately in a similar manner. At the end of the procedure, both groups were kept at room temperatures for 19 minutes. At the end of the incubation period, if the neem extract affects the external structure of virus and its receptors, the estimated time required for this activity to occur was provided. At the end of the incubation period, the cell growth medium was removed from the plates and the wells were washed with PBS. Virus suspensions treated and untreated with neem were transferred to each deletion steps on micro placed in 4 copies in 100 microliter volume per their deletion coefficients. At the end of the process, the plates were transferred to incubator with 5% carbon dioxide at 37 C. The cells inoculated with the virus were checked daily with a tissue culture microscope for the pathogenic effect of the virus on the cell, and the study was terminated at the end of 72 hours to determine the infective dose of a viruses in wells with without neem. A 50% endpoint titer of the virus calculated according to method. In daily examinations under microscope and viability test performed with trypan staining method, no cytotoxic effect was observed in MDBK cell lines exposed to different deletions of neem bark extract for 24 and 48 hours, starting from 1/32 deletions values. In line with the data obtained, the concentrations value to be used in the study was accepted as 1/32. Antiviral studies. effects of Nimbark extraction of BCOV, BHV1, BPIV3 and BEV replications. The lowest non-cytotoxic deletions value of 1/32 of Nimbark extraction was used on BPIV3 and BHV1, BCOV and BEV. Virus replications activated in infected MDBK cas of 1/32 concentrations value were investigated by the method clearly stated above, and TCID 50 results obtained at the end of the 72 hours are as stated in table 1 together with control groups. According to the data we have obtained, it's thought that neem bark extract deletions at a ratio of 1/32 doesn't play a suppressive role of any virus replication on BPIV3 and doesn't cause a significant change in BHV1 and BEV. However, it's seen that the virus infectivity strength is reduced by about 30 times in MDBK cells that metabolize neem extract and are infected with BCOV. In light of the results of research expressed, it's thought that neem extract may play a limiting role in virus replication on BCOV. Effects of neem bark extract on cell surface attachment of BHV1, BEV, BPIV3 and BCOV. The 1/32 neem bark concentrations values used in the replication study were also used in the study, and the study was carried out with above mentioned methodology. And at the end of the 72-hour study, the TCID 50 results of the viruses are expressed in table 2 together with control groups. According to the data we have obtained, it's thought that neem bark extract deletions at ratio of 1/32 doesn't have a significant effects of attachment of BPIV3 and BEV to the host cell. However, it was observed that was a 100-fold decrease in TCID 50 values of BCOV virus treated with neem extract, and virus replication was completely blocked on BHV. Here we see comparative CP images of virus treated with neem and untreated virus. In this study, the ability of NBE extract from Azardiric indic to enter the cell and inhibit replication of BCV, BHV1, BI3 and BEVs after entry was investigated. Tveri et al. Investigated the potential of neem extract to inhibit entry of the virus into target cells, herpes simplex 1 infections from herpes viridi family, in parallel with determination of antiviral activity of neem extract on BHV. Researchers stated that 50 and 100 microgram per milliliter concentrations of neem extract significantly blocked the entry of HSV1 into the cell, and that is blocking activity occurred during the pre-incubation of virus with extract without cells. That is, it was directly effective on the virus. Mahmoud et al. Evaluated the antiviral activity of NB against Newcastle disease virus, which belongs to genus Paramyxovirus, by spot test and microhemagglutination test in vitro. In the in vitro evaluation similar to this study, when the results of no deletions and 1 to 2 deletions of neem bark extract on PIV3 virus were evaluated, it was stated that it exhibited antiviral activity. However, these concentrations also showed cytotoxic activity, but did not exhibit any significant antiviral activity at a higher deletion. In a study, Borkotoy et al., the molecular effects of some compounds derived from neem on the E and M proteins of SARS-CoV-2 were investigated, and it was determined that these compounds have significant inhibitory activity using MD stimulation and binding-free energy calculations. In our study, it was determined that it has antiviral activity against bovine coronaviruses in parallel with researches. Falkendahl et al. Aimed to determine the activity of 2 polysaccharides is isolated from the leaves of I. indica and their chemical sulfated derivatives against poliovirus type 1. Researchers found that polysaccharides did not show any cytotoxic effect on HEP2 cells even at high concentration tested, and that inhibitory concentrations of 80 milligram per milliliter, 37.5 milligram per milliliter, 77 0.5 milligram per milliliter and 12.1 showed significant antiviral activity against poliovirus type 1. They stated that the polysaccharides they obtained from azadirachtin acted against PV1 by inhibiting the first stage of viral application, and that the original polysaccharides showed better virucidal effects than their sulfated derivatives at all tested concentrations. In conclusion, neem is a versatile medicinal plant with antimicrobial, antioxidant, anti-inflammatory, and anti-cancer activities. Phytochemical analysis of neem bark extract was not performed in this study, so its active components were not identified. It said that punicalagin, which is abundant in P. granatum extract and juice, needs bioactivity guided fraction studies. In light with the finds, punicalagin caused loss of infectivity due to its binding to antigenic determinants of BCOV and BHV is thought to be. Compounds other than punicalagin may have inefficient antiviral potential to inactive all infection virus particles. After incubation of neem with target cells for cells to metabolize neem extract, BCOV infections potential appears to decrease by about 30 times. It was thought that neem compounds inhibit the assembly of BCOV particles, thus reducing viral application. Interestingly, complementarily, the blocking activity of neem was seen when only BHV virions were pre-incubations with the extract as compared to pre-incubations with target cells. It's thought that the full elevations of active ingredients in these plants will lead to important path to development of new and effective antiviral agents. In addition, neem anti-herpes activity may be evaluated against individual BHV glycoproteins, fertile toxicity of neem and its characterizations will aid in the development of therapeutic agents against BHV. Thank you for listening to me.
Thank you, dear Pelin. It was quite a race for you. I congratulate you. It's very nice to see young academicians and researchers who are joining more experienced, let's say, not old, but experienced researchers and professors and continue their work. It's very nice. It's very nice to see this. I want to mention that coronaviruses are a large family of viruses, and every study upon them using different plant extracts, using bee products are welcome. And in this study, we could observe that the neem extract is effective for BHV1, which is a start. And I'm recommending for the future the chemical composition, to determine the chemical composition of these extracts, because the compounds which are responsible for a certain property, they are very important, and further studies must be made. But it's a huge study, and I'm congratulating you and all the team which made this. If some questions raised from this study, please put them in the chat. Thank you again, dear Pelin.
Thank you.
And now, last but not least, if Dr. Nimra Mubin it's here with us, we invite her to share the research with what she is. I saw her on the wall.
Dear Professor Otulebabis, she's here, but I think she's not able to open her voice.
Oh, you can,
because I have called them, and she came to us. Nimra, I am trying to open her voice. There is two Nimra Mubin. And I'm trying to call her again.
Okay.
Açıldı. online now.
Okay.
Yeah, okay, thank you.
Okay, Dr. Nidra, can you hear us? Dr. Nidra, can you hear us?
Yes, sir.
Yes, sir,
ma'am.
You have to close because it's a microphony. You have to close one device. You have opened two devices and we hear you with an echo. Okay, now you can try to share your screen with the presentation.
Everyone hear me?
Yes, we can hear you. We are waiting for the presentation to upload. Okay, now it's okay. You can start your presentation when we are ready. Unfortunately, the sound is not good. We cannot hear you.
Yes, ma'am.
No, we cannot hear. No.
Items of fish infected by Aromonas are white film on the fish body are fins, cloudy eyes, striated fins, and hemorrhage are open spores on the body and mouth. Aromonas varronae is a gram-negative rod-shaped bacteria normally isolated from clinical samples of fish, having similar signs just as other species of bacteria. In order to treat this fish, determination of the causing species of bacteria is very important. Nowadays, phylogenetic tree studies and DNA sequencing are getting Aromonas varronae separated from diseased fish to evaluate pathogenicity of Aromonas varronae in Valaguato. Materials and methods. this fish species was used Blackwater, and the sample was collected from fish ponds situated in district Kasur, Punjab, Pakistan. The experimental site will be conducted in Fish Hatchery Unit, Department of Fisheries and Aquaculture at UVA, University of Veterinary and Animal Sciences, Lahore, Pakistan.
Unfortunately, we lost you again, Dr. Nimra. We cannot hear you.
Yes, ma'am. Experimental pathogenicity test. Aromonas varronis suspension 0.1 and 1 CFU per ml was injected intraperitoneally to each fish of the experimental period. Fish were fed with 5% body weight and 25% CP commercially available feed twice a day. After 2 weeks trial, hematology of fish blood and histopathology of kidney, liver, and spleen was done. Statistical analysis, one-way ANOVA was applied for the comparison of different treatment. Deccan multiple range test was also applied. Results, the following pictures are shown of bacterial growth, genomic DNA extraction, and PCR amplification of extracted DNA was done. Phylogenetic analysis of Aromonas varroni. In phylogenetic analysis, Aromonas varroni was confirmed after construction of phylogenetic tree, showing a resemblance with other Aromonas species. These are the four phylogenetic tree you can see here. Sequencing of Aromonas varroni. This sequencing was used for the Aromonas varroni. Final results of DNA extraction. Finally, the bacteria Aromonas varroni was confirmed after the DNA sequencing. Antibiotic sensitivity. Different antibiotics were used to check the antibiotic sensitivity of Aromonas varroni. The different discs which we used, in which penicillium G, tetracycline, gentamicin, oxytetracycline, nobi, biosine, trimethoprim, and neutrophenamine, in which some of the antibiotics are resistant to show resistant against Aromonas varroni. Experimental pathogenicity test. Injecting the Aromonas varroni was injected to fish intraperitoneally. Different bacterial doses were given to fish. Histopathology parameters you can see here. The treatment to which can 1 CFU per ml injection was given to fish. Then the hematology parameters are shown very low results due to the Aromonas varroni. Histopathological analysis. These following pictures are show samples of spleen, kidney, and liver of control and treatments good. They were showing different symptoms on fish. Conclusion. Finally, the bacteria Aromonas varroni was detected after DNA sequencing. Aromonas varroni was sensitive to nitrobiothin, novobiosine, and gentamicin, causing histopathological changing in different organs of fish. Thank you.
Okay, thank you very much, Dr. Nimra Mubin. With some technical problems, we managed to reach to the end. And if you have some questions for from the part where the sound was not very good with Dr. Nimra, please put your questions in the chat. Now we have finished our 15 presentation, very interesting presentation from the field of animal science and veterinary medicine. And I want to congratulate to all the presenters, their research teams, and also I want to thank you, Professor Ulaş Akaroz and Amla, for inviting me to be your co-host today. And I hope that the third Congress will give us the chance to meet in person, to be well in the world. And what I can say is that we pray from our colleagues, researchers and academicians from Ukraine to pass this problem which they are confronting now. Thank you very much again, Professor Ulaş. Thank you very much, Alma, and all others organizers. And see you next time online and in face, if it's possible. Thank you again.
I appreciate your presence and your support, dear Professor Otille Bovis. Thank you very much again for your support. Hopefully in June, we can organize the API Therapy Congress together.
Yes. Yes.
Thank you. See you. Dear respected participants, we will continue in 20 minutes in 18. Then during this meantime, you can enjoy the music and presentation.
Bye.
Şeyi açacağız. Müziği açacağız. Nasıl açıyorduk? Bir an gittiğini zannettim de nasıl yapacağımı şaşırdım. Dur, şöyle yapalım bir saniye. Bir canlı yayını durdurayım ben.